Okay, so this is the PowerPoint for the final exam, the review. So a man is found to have one bar body in his somatic cells. So remember, a bar body is condensed chromatin of the X. Remember, females have two X's, males have one X. And so only in females should we find a bar body, because only one of their X's would be condensed because of dosage compensation, and so we would have equal amounts for each sex from the X chromosome. However, it's possible that you could be XXY, and in that case, your chromosome makeup would be here, and you'd have 47 instead of 46, which would be 2n plus 1. So the chromosomal constitution would be XXY. And then you could take this further and say, what if there are two bar bodies? Well, then it would be XXXY, because these would have to be condensed, or the dosage wouldn't be correct for XY. Which represent a polyploid chromosome number. So remember, poly is many, ploidy is a whole set of chromosomes. So this is haploid, this is diploid, this is aneuploid, and that's +1, and this is aneuploid, and that's -1, and this is triploid. So the correct answer is E. Polyploidy is 2 or more. More than 2. So that would be 3 or more. So 3N, 4N, 5N, etc. would all be polyploidy. Tetrad experiences non-disjunction during meiosis 1. How many of the resulting gametes are nullosomic for a particular chromosome? So we talked about this. Nullosomic means that there's none. So in non-disjunction of meiosis 1, in meiosis 1, remember that the chromosomes travel as tetrads. And so these tetrads would be divided in meiosis 1 into dyads, and then the dyads would be divided into 1N here, which would be the correct number. But in a nullosomic, in a non-disjunction of meiosis 1, you would get 2 tetrads, 1 tetrad, 2 dyads. And this one you'd get none, because both of these would move over to this, and this would get none. So in the end, when this division occurs, we would get none, and then this division would occur, this would get 2. So this would be 2n minus 1, right? It has 1 less chromosome. And then this would be 2n plus 1, because it would get an extra chromosome. We're just not showing all the chromosomes there. So the answer is, how many are nullosomic? Well, this one and this one, which is 1 plus 1, is 2. Non-disjunction of meiosis 2, same deal, right? So we get regular division here. This is meiosis 1. Here, they're supposed to divide down the middle, right? But in one of these, you have non-disjunction, so they both go to 1 gamete. So I've taken the X and I've made it into 2 individual chromosomes like this. So that's why I'm drawing this here. So we have this one, and then this one doesn't get any, so it's 0, and then this one would divide normally, so we would get 1 of each, and these would end up being normal. So how many would be disomic? That means it has 2, and the answer is 1. Only this one here would have 2 chromosomes, an extra copy. So 1, 2, 3, centromere 4, 5, 6, 7, 8. 1, 2, 3, centromere 4, 5, 7, 6, 8. So this would be an inversion, but it is not including the centromere. Remember, this means that it includes the centromere, and this means that it doesn't. So the answer is this is a paracentric inversion. Phenotypically normal fly, large pairing loop meiosis, which is finally, this is most likely to be deletion or an inversion. So, in a deletion, we have, like, let's say we have this is a chromosome, we have A, B, C, D, E, and F. In a deletion, we would have something like A, B, and then E and F. So this would get cut out. So what would happen is that when these paired, you would have a loop, but it wouldn't be, it's more of a bulge than an actual loop. So because this says a pairing loop at meiosis instead of a pairing bulge, which is what this would be. So we'd have A and B, this one, and then this is E and F. This one doesn't have a C and a D to pair with, so it looks kind of like this. So an inversion would be more like this. So we have an inversion. Let's say the normal chromosome is A, B, C, D, E, F. And this one is A, B, E, D, C, F. So when this pairs up, it would end up looking like this. A, B, and then it had to loop around like this. And this would be C, D, E, F, like so. And then the other chromosome would loop over it like this. So it would be A, B. So this is this chromosome here. This one. And it would make this loop right here. And then this one would bulge out like that. So it would be A, B, E, D, C, and F, which is this chromosome here. So this pairing loop would be made by an inversion rather than a deletion. So the answer is B. Can't synthesize threonine. So we want to get a mutant that can't synthesize threonine using replica plating. So if we use minimal media and we have minimal media with threonine added to it, if we grew, so this would be considered the primary plate, and this would be considered this would be the primary plate, this would be the replica plate, because you couldn't grow it on here. So if we plated out all the bacteria on this, on minimal media, and it couldn't make threonine, it was threonine minus, all the bacteria would die. So we can't do that. If we're looking for a threonine mutant, we have to plate it on minimal media plus threonine, and then we would have the bacteria grow, and then we would replica plate them on minimal media, and the ones that needed threonine, they wouldn't grow, they'd die. So then you just compare it back to this plate and say, oh, there's a spot here, but there's no spot here, so that has to be a threonine minus bacteria. So you'd add threonine to the primary plate only, which is the one, not the one you're replicating, which is be this, but the one you're actually growing the original bacteria on. So that answer is A. HFR and E, F minus strains of E. coli. So we can map this. We can map the location of mutants on an E. coli strain. The reason that we can do that is, let's say that we have an HFR and that has a chromosome, and then so in the HFR we have, this is kind of hard to do. Let's say we have genes A, B, C, D, and E, and they're in that order. So when if it splits here and it's starting to pass those across, and this is A and B and C and D and E through the pilus to an F minus strain, what's going to get transferred first is A, right? And then the next one to get transferred over would be B, and then the next one to get transferred would be C, and the next one to get transferred would be D. And so if you interrupted the transfer at each one of these spots, you could tell what the order of the genes are in the HFR and assume that's the same as in the F minus strain here. So you can map genes on the chromosome by this transfer method. We talked about this. So the amount of DNA that gets transferred to the recipient, the F minus cell, depends on how long they're conjugated, in other words, how long they're connected. So if they're only connected for a minute, maybe only A goes across. And if they're connected for 2 minutes, maybe they get A and B to go across. And 3 minutes, so on and so forth. So the amount of DNA getting transferred is directly proportional to the time that they're linked together and through this pilus. B, the recipient cell is converted to a donor cell after conjugation. Remember, it has to transfer its entire chromosome. An HFR has that and that F factor in its chromosome, so it would have to transfer its entire chromosome to an F minus since it cuts in the middle. So it would have to go all the way around all 4 million nucleotides to get back to the other half of this F gene to make this an F plus. So the recipient is converted to a donor cell after conjugation. That's absolutely not true. So the correct answer would be B. All right, we have a partial diploid. The easiest way to do this is just draw out the chromosomes. So I'm just going to do this. We have lac I, and then there's a space in between there, and then we have the promoter and the operator. These are all part of the promoter region, or mostly. And then we have lac Z, and then we have lac Y. All right. So the first one, this is I minus, which means that it's broken, and so we don't get any protein product. So that I doesn't work. This O plus, it's fully functional. Z plus, so that means that if this gets turned on, then we'll make Z. And if this gets turned on, we won't make Y because Y is broken. It could be there's a mutation in it, like a stop codon or whatever, but the protein product of Y is non-functional. That's why it's called Y minus, right? And this protein product is functional because that's why it's called Z plus. So let's draw the other chromosome. Here we have the same ones, I plus. So this one is going to make I, which is the repressor. Then we have the promoter, the operator, Z and Y. So let me just erase this. Okay, so this one is constitutive. That means that this is broken. So if let's say this protein recognizes as the sequence AAA, and this is ATT because it got mutated, it would only be able to bind to this one here, right? It wouldn't be able to bind to this one here. So this one, there's no repressor because this is constitutive, and that means that whatever the product of Z and Y, that mRNA is always going to be made. Now, the question is, does it function? And that is told to you in the question here. So in this one, Z is broken and Y is good. So Y is going to be made all the time. And so we know that Y is going to be there. Plus and plus no matter what. Because this is broken, so there's no repression. It's got a mutation in the operator. So OC means it's always on. Okay. So this one, this inducible system, this repressor is fine, and so is this operator, so it will act normally. So in the presence of IPTG, which was what this is, it means that there's lactose or IPTG in it, then this can't bind, and this will get turned on. So Z will be made, so Z would be plus. And this one, the Y is minus, so even though the mRNA gets made, the protein won't work. So that's out. Now, if it's not induced, then this repressor here will bind. I'm going to clear this up a little bit since we don't really care about this one anymore. And the reason we don't care is because we know it's constitutive. The only thing we care about is that this is I plus. Right? It tells us right there. This is I plus. So it means it makes the repressor, and the repressor will bind to a normal operator, which is this one right here is the normal operator. So they're on different chromosomes. It doesn't matter because this right here, this I plus, is a trans factor, so it means it can move around in the cell and go and bind to whatever it wants. So so if we add lactose, then this gets turned on, so we make Z. If we don't have lactose, then the repressor binds to this. The RNA polymerase can't bind, so it shuts the whole thing down. So neither one of these are made. So this answer here is that if we don't have lactose, this turns off. So the full answer is minus plus plus plus, which is B. All right, we got another one. So IS is a super repressor. A super repressor. So let's write it out again. So we have I, P, O, Z, Y. and then we have I, P, O, Z, Y on the, this is for the first chromosome number 1 here. This is for the second chromosome number 2, which is here. I'm going to write above it. Okay, so this one, this I is minus, so it's broken. It's not going to make anything. This operator is constitutive, so if an I were made, it would bind to this. This super repressor it would bind, it would normally bind here, and it would also be able to bind here. But remember, this has a mutation in it. So let's say that this normally would recognize AAA, and this got mutated to ATT. Then it couldn't bind here. So it doesn't matter if it's made or not. It cannot bind to this constitutive promoter, or operator, sorry, in the promoter region. That means that this can't stop the polymerase from binding and turning this on. So whatever these are, they're going to be on all the time. So this is actually a pretty complicated one, but this one, the constitutive one, shows us that Y is plus and Z is minus. So Z, even though the mRNA will be made, that protein won't be made, and this Y is going to be on no matter what. So it'll be on because it's constitutive. It'll be on if it's non-induced, and it will be on if it's induced, because this repressor here can never bind to this operator, and so the polymerase will always turn this on, and this will always be made regardless of what happens. Now, super repressors are weird because the effector molecule, which is lactose, right? When lactose is present, it would change the shape and cause it to not to bind to this operator. But lactose doesn't stop it. So this, I don't know what happened to my color, but so this repressor, because it's a super repressor, and this operator, because it's a normal operator, this would stick to it. So no matter what, these would get turned off. So it doesn't matter if this is plus or minus, they're both going to be minus. So LAC-Z, because it's broken on the constitutive one, even though it's plus here, this would never get made because that gene would never be able to get turned on because the repressor would always sit there. LAC-Z would be minus if we induced it. And then if we didn't induce it, it would still be minus. That would be the normal state. So the answer is minus minus plus plus, which is D. Yeah, that one's that's probably as complicated as it gets because you're dealing with a super repressor and a constitutive promoter all at the exact same time. So if you can answer that one, you're golden. Which of the following is true of enhancer sequences? So we talked about general transcription factors bind in the promoter proximal. And so general transcription factors would not bind to an enhancer because that's a promoter distal sequence. So that's not right. They can only affect the transcription of upstream genes. Well, no, and we haven't talked about this yet, but we will tomorrow. Enhancer sequences, they can be in the gene, or they can be upstream, or they can be downstream. It doesn't matter, and so they can be anywhere. And so that's not true. They don't have to just be upstream. They can act over very long distances. Yeah, and I told you guys that I would post that. We've talked about SRY, and that makes maleness. And there's an enhancer called SOX, or there's an enhancer for a gene called XOS that's, I think, maybe 1 to 10 million bases away. And it still controls the activity of this gene. So super long distances. That is correct. They can only affect the transcription of downstream genes. No, like I said, it doesn't matter if it's upstream or downstream. Enhancers are independent of where they're located. They can be in an intron. That's true too. So this is an incorrect statement. So the correct answer is C. We'll talk about that tomorrow. Okay, galactose metabolism. Okay, so we haven't really talked about galactose yet, but we will go over this tomorrow. And GAL1 is galactokinase. You guys remember what a kinase is? It adds a phosphate to a molecule. GAL2 is permease. It's the same as lactose, so remember, a permease allows it to enter into the cell. This is galactose transferase, and that has to do with the metabolic pathway. And then GAL10 is epimerase, and all these acting together ultimately produce from galactose the glucose 6-phosphate, which is the molecule, in case you guys forgot, is the first step in glycolysis. So for glucose, it would just add a phosphate at the number 6 carbon, but because it's galactose, it has to do all of this to convert it into glucose so that it can go into glycolysis. So the question asks, which of the following mutations is likely to cause the yeast to turn on all of these genes constitutively all the time in the presence or absence of galactose? You guys know this is the same sort of deal as lactose. So if you have galactose, if there is no galactose present, do you want to turn on the genes to break down galactose? And the answer is no. And then we haven't talked about this yet, but there is a upstream activator sequence, or UAS, which GAL4 binds to. And GAL4 is responsible for turning on galactose. So we have a UAS, and then GAL4 binds to this DNA sequence. So this is a cis sequence, cis, and GAL4 is trans, so it's a protein. And GAL4 acts like a transcriptional activator, so it will turn this on. But GAL80 binds to this and prevents it from activating. So GAL80 is sort of a blocker, and it sort of turns off GAL4. Normally GAL4 would turn on the genes, but it can't do this here. So a non-homologous mutation that destroys DNA binding structure of GAL4. Well, GAL4 wouldn't bind, and GAL4 is required to turn the gene on. So if GAL4 couldn't bind to the UAS sequence, dang it, then the gene wouldn't get turned on. So it says that the gene's turned on all the time, and that is no way. No, X. Non-synonymous mutation that destroys activation of the domain of GAL4. So GAL4 has 2 domains. So this is the DNA. This is the activating sequence. This domain binds to, so this is the DNA binding domain, and this is the transcriptional activating domain. And so it says it destroys the activation domain of GAL4. So this is broken. So what do you think would happen if the transcriptional activator was messed up on GAL4? Its job is to turn the gene on. So it would not turn the gene on. So this would make the gene turn off. A would make the gene turn off. B would make the gene turn off. Deletion in the promoter of GAL3 gene. Okay, so GAL3 is required to knock off GAL80 to turn it on. So it's something else. That GAL3 will bind to GAL80 and knock it out of the way. So if GAL3 isn't there, then GAL80 would always get stuck here. And because GAL80 would always get stuck there, this would get also always turned off. So what if without GAL80 though, if there's no GAL80 to bind here, so this would be GAL80. If there's no GAL80 to bind there, and this transcriptional activator's working, the gene would always be turned on. So this would cause the gene to always be turned on. What is happening? Anyway, that's the answer, is C. IGF2. So this, we haven't talked about this yet. Okay. Well, my eraser's broken. Okay, so we haven't talked about IGF2 yet. I'm going to split this up. So let's say that this is the male mouse. And in males, we have homologous chromosomes. One they got from their dad, and one they got from their mom. And in females, we have the same deal. One chromosome they got from their dad, and one chromosome they got from their mom. So let's say that this first one is IGF. That's IGF2. I just don't want to write out the 2. And this one is H19. Same thing here. IGF2. Fine. Okay, so when these cells undergo meiosis, in the parental copy, the IGF2, the IGF2, whatever that is, gets turned off. And in the end, H19 gets rewritten. So in males, only the IGF2 is active and passed on. And so in the end, so this is dad's chromosome in the dad mouse. We'll just call it a P for paternal. This is mom's chromosome in the dad mouse. Dad mouse. So this is M for maternal. So what happens is these get erased, and then whatever this is gets replicated in both the gametes at the end of, so this is meiosis, and at the end of meiosis in the gametes, whatever this P is the same for both IGF2 and H19 in the parentals. So let's say that this is always turned off and from the paternal chromosome, and this is always turned on from the maternal. And this has to do with well, it's kind of an interesting story, but it has to do with reproductive success of the male mouse and the effectiveness of the female to have genetically superior mice offspring. But I'm not going to get into this because this is already 30 minutes long and it'll take forever to convert to an MP4. So this one is always on for IGF-2 from dad, right? This is the paternal chromosome. And this one is always off from mom. Dang it. Off from mom and on from mom for H19. So that's how this sets up. This is exactly the opposite here. So this is from mom mouse. This is the chromosome she got from her father, so we'll label it with a P. And this is the one she got from her mother, so we'll label that with an M. So in this one, the dad copy of IGF-2 is on in the mom, right? And off in the paternal copy. Because IGF is only turned on in the paternal copy, and it's always turned on in the paternal copy and it always turned off in the maternal copy. So this one is off and this one is on because this is the chromosome that we got from mom. And in this chromosome from mom, IGF-2 is always off and H19 is always on. Same thing here. IGF-2 is always off from mom and always on from mom and always on from dad and always off from mom. Always on from dad. IGF-2 is always off for mom and always on from dad. And then the opposite is true here. So again, this goes through meiosis, and then in the end, it gets gametes. But these gametes are rewritten to be exactly the same as mom's. So this would be off. IGF-2 would be off, and H19 would be on. And then both of these copies, they would be identical. And this one would be like dad's. So this would be like mom's here. This is off. This is on. So these are always on. These are always off. And it doesn't matter what the makeup is here earlier. That's just how it is. Same thing here. So this one is always going to be on. This one is always going to be off, just like it was from dad right here. In both of these. So this is on, this is off. IGF-2 is always on. And it's always going to be the one that it got from its dad. So it's going to copy that. All right, so let's go back since I kind of explained that. And I'm going to erase all this. Let's go this. So we have IGF-2 is essential for mammalian embryos. We know that it has to be on. A male mouse is heterozygous at IGF-2 gene, one wild type allele and one with a non-functional promoter. So we have a male mouse and it has one copy of IGF-2 that's broken. Let's look at X. The other copy of IGF2 is normal, right? So it's heterozygous for IGF2. If this male mates with a female, that's homozygous for the wild type IGF2 allele, which of the following would occur in their progeny. Okay, so we said that no matter what, the copy that this male mouse gets from its father is going to be copied into its gametes. So the first thing we have to figure out is which IGF2 is it from dad and which IGF2 is from mom. So if this IGF2 was from dad, right, and because it copies whatever dad's is, if it was broken, both of these would be broken, and so this mouse would have no IGF2 and it would be dead. So we know that this one has to come from dad, right? And this one has to come from mom because only the paternal copy is turned on, right? The maternal copy is suppressed or imprinted here, so that's turned off. So we know that in meiosis that the paternal copy is rewritten, dad's copy is rewritten for IGF2, and so we would get both gametes would have IGF2 plus and IGF2 plus, and that would cause okay, so let me start this over. What's going on? Okay, so we have, I'm not writing on this side because I'm writing. So we have the dad. And that dad has IGF2 that works. And IGF2 that doesn't work. And one of these is going to be silenced. So we know that this has to be mom's that's silenced, because if it were dad's, if dad's is always active and mom's is always turned off in the imprinting. So we know that this has to be the chromosome that he got from his father, otherwise he would be dead. So these are rewritten. The imprint is turned off. So both of these get turned back on. And so you end up with a normal gamete. And one that's broken. And then meiosis, these are going to separate, right? And so these would be sperm from the mouse. And then it says that he mates with a homozygous wild type IGF female. So her IGFs will be no matter, even though that they are both wild type, so they're IGF plus and IGF plus, because they're coming from mom, the IGFs get imprinted to be turned off, right? And these get from dad get imprinted to be turned on. So these act just like their IGF minus, and these act like their IGF minus. So when you have these, this one comes from dad, you end up with IGF plus and IGF minus. And since this is from dad, this would get shut off anyway. And so these offspring from this sperm fertilizing this egg here, these are two separate eggs, would be normal, right? Because it only needs one copy of IGF, because this is shut down in the mom's copies anyway. This one is problematic because this is broken, but now it's re-imprinted, so it's not condensed, and that will fertilize one of mom's IGFs. Even though it's good, it's still going to be shut off. So this is going to be IGF minus because it's going to be turned off in the imprint process, and then this one will be IGF minus, even though it's going to be turned on because it comes from the dad. So these wouldn't have any IGF produced at all, and so these offspring would all die. And so the correct answer is A. Half of the offspring would die because they wouldn't have any IGF protein. Expression of Drosophila embryogenesis. So maternal effects is the first one. That's like bicoid and nanos. And the gap genes, because those cause large deletions in the embryos. And we haven't talked about that. We'll talk about this when we get to developmental. And then the next one is the pair rules genes. So we talked about like odd and even, and then these are the homeobox genes, which are in charge of actual developing of like wings and feet and legs and things like that. So the correct order of expression is maternal effects, gap, pair rule, and homeobox genes. So the answer is C. Primary role of maternal effects gene. So maternal effects genes are bicoid and nanos, and they set up a gradient that causes head and tail formation. So they're determining the posterior and ventral axes of the fertilized egg. Which I would never say this on an exam, because we didn't even say this word. But this is the anterior and posterior. So butt, head, and then dorsoventral would be the top and the bottom. And it basically, so maternal effects genes set up the major plane for the developing organism. I don't even know why that's a review question. That's kind of a bad one. Okay, so bicoid. BCN is the loss of functional allele for bicoid due to a nonsense mutation, so that's where the N comes from. Male fly, homozygous for BCN, is mated to a female fly, heterozygous with wild type, bicoid, and BCN. Alright, so we just do the cross. So here's the male. This one has homozygous for BCN. Okay, so BCD bicoid N, which is nonsense, which means it's broken. And this is B, C, D, N. So both of these are broken. And the reason that this fly can exist is because it's a maternal effects gene. So it wouldn't have either functional copy of bicoid. So if it didn't have a functional copy of bicoid, it wouldn't be able to develop into a normal fly. But remember, the mother is what sets down the nurse cells for bicoid. So as long as she has a functioning copy of bicoid, then she will have nurse cells. Even if her offspring are bicoid defective on both, they can still develop normally. Because it's the mom's genotype that's important, not the embryo. So male fly homozygous for this mutation is mated to a female fly that's heterozygous. So female has BCDN and then normal bicoid. So this fly can only make sperm because it's a male with BCDN. We can do a Punnett square to show this. This is mom, so she's BCDN and then BCD plus normal. So this one would be this mom is heterozygous for wild type bicoid. So she has enough bicoid to actually have all of her offspring develop normally. What's the following best prediction for the offspring phenotypes? So we don't even have to do this cross. I don't even know when I started this. All we need to know is that mom has bicoid. And if she has bicoid, then all of her offspring would develop normally. Oh wait, we do need to do the cross because we need to know what her offspring's offspring would do. So all of her offspring would develop normally. Because bicoid isn't contributed by males, it doesn't matter what the males are. So we do BCD N. Both copies are bad. If this is a male, it wouldn't matter because he doesn't contribute any bicoid because this is a maternal effects gene. If it's a female, then all of her offspring would be dead because she couldn't contribute bicoid, so they wouldn't get a normal head and tail. If this is BCDN and then plus, this would mean that all the males doesn't matter, right? And for females, because they have one good copy of bicoid, this female's offspring would develop normally. So half of the female offspring cannot produce their own offspring that develop into adults because these females are homozygous for bicoid NN. This one is heterozygous. It has one plus and one N. So all of her offspring, because of this, would develop normal heads and tails. All of this female's offspring would die because they couldn't develop head and tail. So one half of the female's kids would be dead. One half of the other female's kids would be normal. So the answer is C. Contains a homeodomain. So homeodomains are always transcription factors. I say always, but of course someone will prove me wrong, but that's what they do. So the answer is B. That's just a memorization thing. We replace one body part for another. could be gain of function or loss of function. Answer is true. We talked about it, like, even though that you lose an antenna and gain a foot, that's still a gain of function because the gene is normally repressed in that head. So it's repressing growing a foot, and that's where you get a default as an antenna. If it repressed where the feet were, then you get antenna for feet. So it's still a gain of function because the gene would normally be turned off, but here it's turned on. It would normally be repressed, but then it's not. So the answer is A. And that's just sort of another memorization question. 2 homeotic genes, SN1 and SN2, are found in newly discovered snail species. SN1 is more similar to Antennapedia than to any other Hox genes in Drosophila. SN2 is most similar to ultrabithorax gene, which is not true. Okay. Not true. SN1 and SN2 are likely to be similar to those of Antennapedia and ultrabithorax. So SN1 is close to Antennapedia, and SN2 is ultrabithorax. Not true. Well, SN1 and SN2, these are homeotic genes, and this is for the Antennapedia in the head, and then this is the thorax in the thoracic region. So these are related to one another, and these are all probably similar because they all have similar sequences. All of these homeotic genes have similar sequences because of duplication events. So this is true. So it can't be that because it's not true. SN1 and SN2 are likely to be next to each other on in the snail chromosome. So Antennapedia and bithorax are on the same chromosome, and so we would think that SN1 and SN2 would also be on the same chromosome, and so that would not be not true. Remember, these are the ones that are similar, right? These ones that derive the same. Remember, we talked about the groups. So if they're in the same group, if they're similar to each other, remember we talked about this with like alpha hemoglobin. And then like we could make these similar to beta hemoglobin. So if we're talking about SN1 and Antennapedia, then those are paralogs, and the answer is no. Remember, things that are similar are called orthologs, like all of the genes that are similar to alpha hemoglobin or all the genes that are similar to beta hemoglobin would be orthologs. So SN1 and UBX, these are paralogs because they're like alpha and beta hemoglobin. So this is true. This is not true. What's going on? This is not true. So the answer is C. Alright. Let's just show you the answer. And that's another slide to show you that. Okay. So female is homozygous for BCDN. It's mated. And so remember, this female, she can be BCDN and BCDN. and BCDN and still develop normally, because her mother could be heterozygous, right? And her father could be heterozygous too. And so she would get this copy from her mother. Her mother would also have a wild type copy, and then this copy from her father. He could be BCDN on both alleles. It wouldn't matter because he doesn't really contribute to the developmental part of the offspring. So anyway, that's neither here nor there. This female has homozygous for BCDN, which is the null-somic. Alright, so male fly is heterozygous, so we can just do a Punnett square. We have BCDN from this female. That's all she can produce, so we don't have to write it in here. And then this male would be BCDN, and then BCD plus. So these would be NN, and these would be N plus. Same thing here, NN and N plus. And we could say that these are the males and these are the females here. So what's the following prediction for the offspring phenotype? So just their offspring, not their offspring, I guess. We didn't really need to do this cross because we didn't know their offspring. This is the mom. She's homozygous with this, so all of her offspring will die. There's no question that asked about her offsprings because they're all dead. So I guess I just did a bunch of extra work for no reason. Parallel gene encodes a transcription factor to a regulatory sequence of which gene would this protein bind. Transcription factor. So remember we have maternal effects, and then gap, and then and then we have the pair rule here. And then we have the Hox genes here. So we have maternal effects, pairs binds to gap, that binds to pair rule, that binds to segment polarity, that binds to the homeotic genes. So if this is a pair rule gene and it encodes a transcription factor, it's going to bind to the next downstream element from that, which would be the segment polarity gene. So that answer is C. Which the following is not likely to activate the transcription of a eukaryotic gene. Okay, so we haven't covered this yet, but acetylation of lysine residues, this is normally positively charged. So it has a high affinity for DNA, which is, as you know, negatively charged. So when you acetylate lysine, you take away this positive charge. And so that causes it to not bind to DNA as tightly, and so that opens the chromatin. And that increases gene expression. So acetylation of lysine residues of histone tails would activate transcription. So that's not right. Binding of the transcription factor to the promoter. Well, we know that transcription factors help turn on or activate genes, so that's not right. Methylation of cytosine residues in the regulatory sequence. So this is actually not likely to activate transcription because methylation turns off transcription, where acetylation turns on transcription. So the correct answer is D, recruitment of the chromatin remodeling complex. So that's just the switch SNF. And this one actually turns off genes. Turns on genes because you're remodeling the chromatin to access the Tata box. So this isn't true. This would activate a gene. So this is the only one not likely to activate a gene. Right, paternally imprinted paternal copies are silenced. So we talked about IGF2 and H19, and we talked about the dad and mom chromosomes. So the mutation causing short H19 protein is rare among mice. Okay, so finds the best analysis of the inheritance of this mutation. Sorry, I got interrupted. So, alright, don't erase. Okay, so here's the deal. We draw out dads. Here's dad. Paternal. And he has a copy of a chromosome he got from his dad and from his mom. This is wild type, so this is H19 plus, and it looks like a G. And this is also H19 plus. And then this is mom's. So she's got a paternal and she's got a maternal copy. And one of these is messed up. So when H19 is wild type and the other H19 is short. Okay. So it doesn't matter what comes from dad because that's going to get turned off anyway because H19 from the father, the paternal copies are silenced. So only these count as part of the gametes. So if these all are individual gametes, we could write out the cross. We could say this is dad here, this is mom here. So this would be H19 plus, H19 plus from dad, and then H19 plus from mom, and H19 short from mom. And so this one doesn't matter, and neither does this, because dad doesn't count. So whatever they get from mom is what they get, what they're going to express. So half of these would get wild type from mom, and half of these would get the short version from mom. So half of the offspring of the short embryo would express the short H19 protein, this half right here, and then half would express the regular length H19. So the right answer is B. And this just shows that. Although polyploids are polyploids created by hybridization of, remember we talked about this, individuals from different species or make allopolyploidy. If it's the same, it's auto, so the answer is B. Klinefelter is associated with individuals that possess, so Klinefelter is X, Y. So they have 6 chromosomes, which is 8. Bacteria that require outside nutrients for growth are known as auxotrophs, just a definition. Integration of the F factor in the bacterial chromosome. Remember, if F goes into the bacterial chromosome, it makes an HFR. That's the inducible operon. The free repressor binds to the operator. Remember, the repressor binds to the operator. Example, this is the lac operon. Positive control required to turn on expression of a structural gene. So that's the product of a regulatory gene called an activator. And if you go back to the slides from today, we talked about that. Tripoperon, structural genes are being transcribed only when the tryptophan level is low. So the tryptophan operon it's a negative repressible operon because when we talked about the definition of negative, and negative is when we have an operator that binds a repressor and then we get no transcription, or we have an operator that has no repressor present and we get transcription. So both of there's a negative, and in the tryptophan operon, remember that there's no repressor present, and so the operator is free for to not have anything bound to it, and so transcription occurs. So that's negative, and then it's repressible because if we increase the level of tryptophan, it decreases the level of transcription, thus repressing the whole operon. So the answer is that it's a negative repressible system. Okay, so second level, we talked about this as attenuation. And remember, attenuation is when translation has an effect on transcription only in bacteria and only because it can occur simultaneously. And we talked about for the tryptophan. These are hormones. We're going to talk about this tomorrow. Hormone regulation. And let's get something. Okay. Steroid hormones they're small, lipid-soluble molecules because they're steroid hormones, not protein hormones. They are all synthesized from cholesterol, and they are all able to pass through cell membranes just like cholesterol. So the answer is D, all of these. Peptide hormones, they don't ever enter the cell because they can't. They interact with membrane-bound receptors, and we'll talk about this tomorrow. So the answer is peptide hormones, which is B. DNA sequence function position independent rate. No, you guys should know that. Those are enhancers. Gene is marked in some way that it remembers where parent it's came from is imprinted. We just did a whole bunch of problems on that. False. Remember, Drosophila do hyperactivation of X so that the males get twice as much dose as the females. So in humans, this is shut down. In Drosophila, these stay on. In this one, the genes get expressed to twice the level. One body part to look like another. So that homeotic mutations, remember the antennapedia, and we talked about this, but we'll talk about this in developmental genetics. In the antennapedia mutation, an antenna looks like a foot, so that's a homeotic gene that controls that. We're done? All right, that's it. So how do I stop this?